In this video, we learn about phototransduction. Phototransduction is a physiological process by which light is converted into electrical signals in the retina of the eye. For phototransduction, we need photoreceptors, that is your rods and cones present in the retina. Main function of receptors in any part of the body is to convert one form of energy into another form of energy. That is like they are like transducers. They convert one form of energy into another form of energy. For example, if you take a touch receptor, the touch receptors convert a mechanical energy into an electrical signal. That is your action potential in the neuron. Similarly, now in phototransduction, when we see in the retina, the rods and cones which are present in the retina, they convert a photon or a light energy into an electrical impulse in the neurons in the retina. So that this process is called as phototransduction. To understand phototransduction better, let us slightly look into the structure of rods and cones. So in this diagram, what you see here is on the left side is a rod, on the right side what you see is a cone. This cream colored portion is your outer segment of the rods, and the remaining portion is the inner segment of the rods. In the outer segment of the rods has been enlarged on the right side here. What you can observe here is there are membranous discs. These membranous discs, they have these molecules present, right? Those molecules are called as rhodopsin. It's a photopigment, okay? So when a light strikes onto these rods, it is the photopigment which are present here, that is your rhodopsin, which actually detects this light energy. Clear? So these membranous discs, if you see, they are separated from the cell membrane of the rods. But in cones, they are just an invagination of the membrane. So as you can see here lower down, no? If you can see clearly, the membrane itself has folded out. So this is how the discs will be in cones. So that's why they usually call it as it's like a sacules kind of thing, okay? But in rods, what they tell is mainly these discs are separated from the cell membrane. Before we understand phototransduction properly, let us learn 2-3 concepts and then go to phototransduction mechanism, okay? Now, I told there are photopigments present in the membranous disc, that is your rhodopsin. So, rhodopsin is nothing but it's a combination of 11-cis retinal and scotopsin molecule. So, when light falls onto this rhodopsin molecule, it undergoes series of changes like bathorodopsin, lumirhodopsin, metarhodopsin 1 and metarhodopsin 2. So finally the metarhodopsin 2, which is formed within few seconds after the light strikes this rhodopsin molecule, is nothing but an active form of rhodopsin. So initially rhodopsin was just a combination of 11-cis retinal and scotopsin. Now once light strikes onto this rhodopsin molecule, what happens is the 11-cis retinal present within this rhodopsin molecule converts itself into an all-trans retinal. Okay, but the combination of all-trans retinal and scotopsin is unstable. That is why they dissociate. Clear? But within that few seconds of in that combination form, no, it is called as an active rhodopsin molecule. So just observe the diagram here. This is a 11-cis retinol with a scotopsin. So together we call it as rhodopsin molecule, right? So this is your rhodopsin molecule, 11-cis retinol with scotopsin, right? Now you can see the protein holding this 11-cis retinol. But when light strikes onto this molecule, 11-cis retinol changes itself into an all-trans retinol. The angulated structure now becomes a straight structure, right? So now the scotopsin molecule cannot hold this all-trans retinol, so it dissociates. Clear? So now you can understand this molecule, we call it as metarhodopsin 2, which is an active form, but within few seconds it separates out. That is what has been showed in this Wald cycle. Clear? Now let us understand another concept here. When the light falls onto this retina here, just observe, light travels through cornea, then it passes through this lens, vitreous humor, and then strikes onto the retina. In the retina, if you observe, when the light is falling like this, no, the direction of light is from the light is passing through the ganglion cells, bipolar cells, and then passes through rods and cones, and then goes and strikes your pigment epithelium here. So this is the direction of light. But the direction of neural communication or the impulse how they travel is, you can observe here, this is your rods and these are the 3 cones represented here. So once the light strikes onto the rods, there is electrical impulses generated in the rods that will be transmitted to bipolar cell, that transmits to ganglion cell, and the axon of the ganglion cell is the one which forms the optic nerve. What you see in this diagram is, this is the rods. There is cyclic GMP gated channels which are shown in the outer segment of the rod. Inner segment of the rod has potassium channels and also sodium potassium pump. Now, in the dark, these cyclic GMP gated channels are open. It will continuously allow sodium ions to enter into the outer segment of the rods, and from the sodium ions from the outer segment diffuses into the inner segments and will be pumped out by the sodium potassium pump. So three sodium ions are pumped out in exchange for two potassium ions. So now these potassium ions, they have lot of potassium sensitive channels in the inner segment, will efflux through that. So this will allow the current to flow from the inner segment to outer segment, right? So the outer segment is allowing the sodium ions, and the positive ions are sodium ions are pumped out by sodium potassium pump, and the potassium which has entered into the segment is going out through the potassium selective channels. Now, because of the influx of sodium ions, this receptor is depolarized and it will be releasing the glutamate. Okay, remember, in the dark, sodium channels are open and there is influx of sodium and there is release of neurotransmitters. But once the light strikes, these channels will close and it will lead to less release of neurotransmitters. So always understand that in any other receptors in the body, usually when a stimulus is given to the receptor, it undergoes depolarization. There will be influx of some sodium ions or any positive ions and it will lead to depolarization. But in rods, in contrast to that, other receptors, here what happens is, when in the dark there is depolarization, there's a continuous release of neurotransmitters glutamate. But once light strikes, that is your stimulus is given to this receptor, it undergoes hyperpolarization. Clear? Now we'll understand another important mechanism here to understand phototransduction better. Okay, cyclic GMP guanylyl cyclase system. This is one of the second messenger system in the body. Okay, many of the cells utilize the second messenger system. You can see here a cell membrane. Gray lines represent the cell membrane, and you have a transmembrane receptor present here, which is linked with a G protein. G protein is nothing but it has a alpha, beta, gamma subunit, and the alpha subunit is attached with GDP. Now, this receptor here, if it combines with an hormone or any stimulus is given to this, what happens is the GDP which was there along with the alpha subunit, no, now GDP will be removed and a GDP molecule will come and bind to that. So the GTP and alpha combination, what happens is it's a highly energized combination. So it separates from beta gamma subunit. So the initially the all the three subunits were together along with GTP, right? But now once the GTP comes and binds, the GTP alpha separates, it will bring about many physiological actions inside the cell. Similarly, beta gamma also have so many mechanisms to be done inside the cell. So now this GTP alpha combination, let us call it as transducin molecule in rods. So this mechanism, what is happening inside the rods, no, that we call it as now like this cyclic GMP guanylyl cyclase system, no, slightly to modify this to the rod mechanism in the rods. What happens is this alpha GTP combination, now let us call it as transducin molecule. Clear? Hope you guys have understood here. This was in a resting state. When a hormone binds, the GDP was bound, GDP was released. Now GDP alpha will bring about many physiological actions, right? Now let us understand here. Now again come back to the outer segment of the rods. So this is a one membranous disc which is shown here, and there is one rhodopsin molecule. So once the light strikes, we all would have understood now in Wald cycles, the rhodopsin gets converted itself into a meta-rhodopsin 2 molecule, which is an active form of rhodopsin. Now this rhodopsin activates a transducin molecule, G protein. So transducin is nothing but what we saw in the previous slide, right? The G alpha and your GTP molecule together. So it is nothing but it's another G protein, that is your transducin, is activated. Transducin activates an enzyme called phosphodiesterase. PDE is the short form. This phosphodiesterase breaks the cyclic GMP. If you remember, in the outer disk, we had a sodium channel which was continuously kept open by cyclic GMP. Now, an enzyme is activated to break down cyclic GMP. Once the cyclic GMP levels comes down, the sodium channel closes. So just understand here, once the light strikes onto the rhodopsin, we get what is called meta-rhodopsin 2. This meta-rhodopsin 2 activates transducin molecule. So transducin activates phosphodiesterase. So phosphodiesterase is nothing but a cyclic GMP breaking enzyme. So cyclic GMP gets converted into 5' GMP. So the level of cyclic GMP reduces inside the outer segment of the your rods. So once the cyclic GMP levels have reduced here, once light falls onto these rods, this channel closes. Sodium will not enter into the outer segment. So what happens is there is no depolarization, or it causes what is called hyperpolarization. So once the stimulus is given, the rods undergo hyperpolarization. So just to summarize again, once the light strikes onto the rhodopsin molecule, the rhodopsin converts itself into metarhodopsin 2 that activates your transducin molecule. Transducin activates phosphodiesterase. Phosphodiesterase is a cyclic GMP breaking enzyme. So it converts cyclic GMP to 5-GMP. And once the cyclic GMP level reduces, the sodium channels in the outer segment closes. And that leads to hyperpolarization. So the influx of sodium is reduced. That leads to hyperpolarization of the rods. So light hits rhodopsin molecule. The 11 cis retinol becomes all trans retinol. The rhodopsin becomes what is called metarhodopsin 2. This is the active form of rhodopsin. Metarhodopsin 2 activates your transducin molecule. There is nothing but a G protein which is present in the rod. So this transducin activates phosphodiesterase. This breaks down the cyclic GMP, so leading to reduced level of cyclic GMP in the rods. Lower cyclic GMP causes closure of sodium channels. Sodium is channel also allows little bit of calcium ions also. Okay, so there is hyperpolarization of the cell. So there is an influx of positive ions has been reduced inside these rods now, which has led to hyperpolarization of the cell. So hyperpolarization of the photoreceptors leads to reduction in the glutamate release. So once the light has striken the rods, remember the amount of neurotransmitter released is reduces. So that change is detected by the bipolar cells that will in turn signal the ganglion cells. So finally the signal is transmitted via the optic nerve to the brain. Finally, the visual information is processed in the visual cortex. In the next video, I'll explain how exactly the image is processed in the bipolar cells and ganglion cells. So to put it in a simple terms, the photoreceptors, your rods and cones, so they detect the light, and once they are stimulated, a signal is created that is sent to bipolar cell. Bipolar cell is synapses with the ganglion cells. Axons of the ganglion cells forms the your optic pathway or your optic nerve. If you have understood the mechanism of phototransduction, please subscribe to this channel and also share it to your friends. Thank you.